Hey Sam fans, and welcome to this biology edition, my first biology edition of Sam Science. So this topic we're going to look at is transcription translation. This is only for triple and Edexcel exam board. I know it's not for AQA at all. I'm not too sure about the Welsh exam board or the OCR, so please do check that. I put a little picture there of tears because transcription and translation is a topic that a lot of students do struggle with, and it makes them, well, brings them to tears. Let's be honest here. Okay, so transcription translation, let's crack on. So a quick sentence description of what transcription is. It's a process by which mRNA, which is a messenger, is formed using the code in a DNA molecule, which we're going to have a look at in a moment. So transcription, how does that happen? It's basically triggered by chemical messengers that enter the nucleus, which remember is where the DNA is found, that binds onto the DNA at the particular relevant gene point. Okay, so the very first stage that you need to know, this usually does pop up as a 6-mark question, which is horrible. So particular enzymes called helicases, they come along and they pull apart the structure of the double helix DNA. Okay, so as you can see there, there's the double helix being pulled apart there, and you can see here it's revealing the bases. So just imagine you've got like a zip up coat and you pull down the zip on the coat, the zip comes apart on the coat, doesn't it? Yeah, just imagine that's what the helicase does there. It's unzipping and opening up that double helix DNA to reveal the bases. Okay, then another enzyme comes along called RNA polymerase, and that then begins to create mRNA, which is messenger RNA, but you can shorten it to mRNA. Okay, the RNA polymerase transcribes, so it literally just copies the bases on one strand of the DNA to make a complementary molecule of mRNA. So RNA basically is making mRNA, which is a messenger, and it's transcribing and copying all the DNA from the bases from itself then onto the mRNA. Okay. Meantime, okay, so DNA, yeah, do you get this? DNA is double helix, RNA is a single strand. See, like double helix, DNA, RNA, single strand. Oh, Sam, you're funny. I know, had to be done. Had to be done. Okay, another important part of transcription is that where there's an A, it is replaced by U instead to the mRNA. Okay, so if you look here, you should know your bases by now. If you don't know your bases, you need to go back and go over them bases. So if you look at the bottom there, you've got your A to G, etc., etc., A, T, G, C, etc., and you know that the complementary bases do match up to it. The only difference is where you see an A at the bottom, the U matches up with it, but if you look here where there's a T, the A will match up with it. Okay, so the U will only match up with the A, not the other way round. Okay, and the U stands for uracil. All right, and that's added to the mRNA. So T is completely gone. Okay, on the mRNA, it doesn't exist on that. Okay, and then finally, what then happens is that the polymerase then stops copying, and as a result, the mRNA is then released out. Now, it's important to know that it leaves the nucleus and it enters into the cytoplasm. Okay, so leaves the nucleus and enters into the cytoplasm for translation. So in a nutshell, transcription is the DNA being pulled apart and the enzyme RNA copying and making mRNA ready for that copying to then go on through to translation. And remember that A is replaced with U at this point. Then translation is, here's a definition here, is a production of a protein. So this is where protein synthesis takes place using the information that mRNA got in the first stage of transcription molecule. So the first stage is mRNA getting that copy, getting the information. Translation is then inputting and putting it together, okay, for making the DNA. So at this point, so remember this is all taking place in the cytoplasm, okay. And the ribosomes as well. So mRNA moves from the nucleus to cytoplasm. The ribosome attaches to the particular sequence of bases on the mRNA. All right, so the ribosomes is where the protein synthesis takes place. So this takes place in the ribosomes. Then it moves along the mRNA 3 bases at a time. Now, the 3 bases at a time I've highlighted here in this picture. In this case, it's A, U, and G. The 3 bases here are called codons. Okay, And there's another substance called anticodons, which pair up with the codons, which I'll show you later on. Okay, so just to go over that again, ribosomes attach to a particular sequence of the bases. Okay, then the ribosomes then move along 3 bases at a time, which are called our codons. Okay, and the opposites that match up with it, because remember, it's got still have its complementary bases. They're called anticodons. Okay. Then next stage here, as you can see here, I've drawn a black arrow there. So molecules known as tRNA this time bring individual amino acids to the ribosome to be joined together as proteins. So this sort of circle, funny shape here with these letters on. Remember, this is the bases here. It's been copied. This whole structure here in my arrow is pointing to is your codon. And you can see the highlighted 3 yellow here, okay, is your codon. And the anticodons is where this black arrow is coming out from. So the circle highlighted in blue with the black arrow, those 3 bases are your anticodons, and the anticodons are going to match up with the codons, which are on the tRNA. So it's almost like it matches up, and then it then moves on and flies off. Where does it go? It just goes into the body, and the process keeps taking place all the time. Okay. Gosh, I had to chuck a meme in there. If I had a choice between DNA and RNA, I would choose RNA because it has U in it. You get that? U, because uracil, and uracil is in RNA and mRNA, and it's not in DNA. Oh, Sam, you're funny. Let me go back for a moment. This is really important here that we know that every time the codons and the anticodons match up, this is when it then creates its polypeptide chains. Okay, so polypeptide chains are small chains of the amino acids. Okay, once they match up there, a massive chain is then produced. Okay, and then this is where you get this protein synthesis in the ribosomes. Okay, so once they match up, the actual protein itself is made, and then the next protein is then made, and then the next protein, and then the next, so on and so forth. So it ends up making like a long polypeptide chain. Okay, and then that's pretty much the end of translation. So translation is basically having the copies of your bases and your information, etc., and then matching them up with the other bases within the ribosomes where the actual protein itself is being made, and that's where your amino acids are also made, then your polypeptides. So this massive chain of polypeptides is then made because it's got copies and copies of your DNA from transcription. Okay, what I have done is not in the comments, in the description, I've put a really simple and easy video to watch. It's got no narration, but it's a very simple, easy video to watch, and it basically shows you the whole process of transcription translation from start to finish. So if I put that in the description, please do have a watch of that. It's a really, really good video to watch, just to consolidate what my PowerPoint was showing. I really hope that that's been helpful for you, and don't forget to subscribe and of course like my videos.